we are going to now yesterday we have seen the cluster W for multiple sequence alignment, so that we can find what are the conserved region or what are the characters, how the characters are aligned over there. And we have done with amino protein sequences and as well as the nucleotide sequences. Okay. I told you, okay, there is a conserved region, partially conserved region, or partially conserved. Now we will talk about the nucleotide over there, or one individual, one column. In that column, if it is completely conserved throughout the intro, from one sequence to the last sequence, it was a star given. So there is no change. But if you see other columns, okay, where the colons is given or just a dot is given, what it tells you that during the evolutionary period, the one amino acids or one residue, one nucleotide has been changed to other nucleotide, or one amino acid has been changed to other amino acid. So can I predict this? What is the chances? Can I use these data for making some models or a probabilistic model or statistical model, just that will tell us what is the rate of evolutionary or what is the rate of change of one character to other character or one amino acid to other amino acid. That can be done. Just we can calculate. Take the examples. We have taken a 20 amino acid, 20 sequences, align them. Okay, you've taken a block, conserved block, which is highly similar. If you find, okay, fair similarity is there, go one by one column you have taken, second column, third column, fourth column. I don't have a multiple sequence alignment here. But you just imagine, you have done that work, you have done a lot of times, phylic format, clustral format, and you know the columns. I told you the conserved region. So one column, second column, third column. Take the one column, what is the probability that one amino acid is replaced by other amino acid? Which amino acid is occurring majority times? Okay. And which is amino acid that is conserved throughout there? So that can help you to estimate. Means if you have two sequences highly diverged, take the example of chymotrypsin and the subpelicin. One is bacterial origin and one is human, higher organism origin. Both are proteolytic enzymes. If I align them, so they are distantly related, having 30 to 40% of similarity, but they are conserved regions. They have the conserved region, means they have active catalytic side is same, means that cuts or recognize the peptide bond and cut over there. So I can take that example, such type of sequences and form the model. Okay, what is the substitution? Means I can form the substitution model over there for amino acids or protein sequences. What is the probability of one amino acid will be get replaced or mutated by other amino acids? And same is the case of what? Nucleotide, because three nucleotides they form one amino acid. Okay, so same is the case for nucleotide. So that helps these, means because we are talking in terms of now we are going for substitution scoring matrix. Okay, today yesterday we have seen, okay, we have generated a file. Now today we are going for actual phylogenetic analysis. What the data given by cluster W was dendrogram got. You just observe that, but it is just a crude phylogenetic analysis. We need to go for a further analysis. Further means we want a replication of the data, and then we can generate a number of tree, and out of number of tree we need a tree which satisfied our criteria. So that is the purpose. So there are, today I am dealing with what are the methods in phylogenetic analysis which are used, and that are used, which are commonly used, and are found most of the papers they refer these methods. Okay, that I am telling you. I won't go for a mathematical background of these methods. It will take a longer time, but I will just tell you what is the basic principle behind the generation of that particular method. Okay, what is the principle of that method? That I will tell you, so that you will get a fair idea. Okay, which method is suitable for you. Next. So computational methods in phylogenetic analysis. Tree construction method, there are two methods. Base means one is either on the one is based on evolutionary distances or a distance method, and second is character method or character state method. Distances are calculated from a pairwise comparison of sequences forming a distance matrix which is used in subsequent analysis. Yesterday, cluster W of run, you got a multiple sequence alignment. Actually, the basic of the multiple sequence alignment is the pairwise alignment. I told you there was file given, input file, output file, okay, then alignment and multiple dendrogram. So there is alignment score filing or output file was also given. So that is a scoring file. And some has observed that file. See what is the comparison 1 to 2, 1 to 3, 1 to 4, 1 to 5. That was given. And then 2 to 3, 2 to 4, 2 to 5. So that is actually similarity score. So can I convert? As I yesterday also in lecture, you got that. So okay, similarity plus dissimilarity. That value, if a fraction value we have taken, then it is equal to 1. Okay. So if we want to know what is dissimilarity value or dissimilarity coefficient, so 1 minus similarity coefficient, that will be equal to dissimilarity coefficient. So that value, score value will be used to calculate the dissimilarity value. And that dissimilarity value we term, or that can be as a distance. Okay. For phylogenetic analysis, we must have either similarity or either dissimilarity. Based on a similarity or dissimilarity, we can group the organism or discriminate the organism. If we consider the dissimilarity, then we can discriminate the two groups of the organism or two groups of the objects. Similarity, that will group the number of persons or individuals in a particular group. So distances calculations of similarity score will be used to calculate the distances among different taxa. I am just taking the term, using the term taxa or genus of species. Okay. Clustering algorithm. The next part is, once I got the distances, what can I do? Clustering. I need to cluster the organism. I know that, okay, the species or taxa A is at this distance from taxa B. Then taxa A is at this distance from taxa C. That is sequence 1 to 2, 1 to 3, 1 to 4, like that. Now next is how I can cluster them. So I will find my criteria is, okay, which are the closely related and which are the, means from A to B and A to C, which are closely related. If A to B is having the high low value of dissimilarity coefficient or lower distances, then I will place them closer to each other, and C will be placed apart from, at a far distance from them. And if D and E is there, the same criteria will be used. Then popular distance methods are like UPGMA, unweighted pair group method using the arithmetic mean, and neighborhood joining method. Both, this was given by Sokol and Smith in 1963 and this was proposed by Saito and Nee 1987. This is the latest one. Both the methods are widely used and distance matrix method is most preferred. Okay, but for systematic, next method is important. Next. So character state method, there are 4 methods. Its main is attempt to fit the characters, nucleotide or amino acids observed in all spaces at every size to the tree. They try, means this is based on what are the changes of the character, means the character which is present at particular position. Okay, and same position, what is the character present in the another species or another object. So based on that, a number of tree will be generated, and from that tree, which is the optimum tree, that will be selected. It's the same like dynamic programming. You have the number of options or a number of possible solutions are there. So same is the case. Number of possible trees are there, but which is the best tree, that will describe the evolutionary relatedness or evolutionary relationship among the different tags or organisms will be the best one. And same is the case. Means like maximum parsimony is the method given by Fish in 1979 and Hutting in that was modified. It's based on a minimum number of changes. Means the tree, okay, for one side it's possible, okay, 3 trees are possible over there. So which tree describes minimum change or minimum evolution? Can say, means A to T or A to G or A to C, if nucleotide model or nucleotide sequences are there. Or amino acids 20 possible. So minimum number of changes, that tree will be selected. Okay. Then second is maximum likelihood method or ML method. It's mainly first instance in 1981, then modified in 1984. He has given two distance matrix calculation also. Models he has proposed, and that we have to use today. His software Philip we are going to use today for distance matrix method, maximum likelihood as well as the parsimony. Both these two we are not going to use. Okay, that are not used widely. Log likelihood score optimized for the branch length and model parameters. It's based on a minimum. Parsimony method is based on a minimum number of changes. As you see, the alignment is there. Okay, in alignment, one column. You've seen the alignment yesterday. One column completely conserved. So it's not telling you anything regarding the discrimination of the group. It will place your all the objects or all the organisms or tags in one group. That shows the similarity. But what about the case where the colon is given? Means out of 10 sequences, 5 are having alanine, and 10 sequences out of 10, other 5 they are having valine over there. That's a colon means, but both are physiochemically similar. Functionality is there, functional relationship is there. That means that tells you you can now divide the groups. Means you can divide that whole taxa into two groups. 5 in one group with valine at that position, and 5 in another group having alanine at that place. Okay. If now go further, another side, another column where 3 are having one amino acid. Okay. Consider alanine is there. 3 are having valine, and remaining 4 are having isoleucine. So you can make a 3 groups for that. So in this way, this is the site. Means it will generate one tree showing all at one place. Then second position, it's telling you 2 groups, 1 mutation. Third group telling you 3 groups. So which is possible? 1 mutation, because first site is not informative. Okay, we will come to this point also. Means when the character is completely conserved in that column, it is not telling you or give an idea to discriminate the organism. But the second position where 5 means one group is we can divide the whole group into 2 groups, subgroups. So that is the informative